catalyst also we define as those chemicals which increases the rate of a reaction without itself undergoing any changes. Okay. Same thing is the definition with the enzymes. The difference is that the enzymes, they are present in the biological system. Okay. So that's the main difference. Otherwise, the function of the enzymes and the catalyst is same. Later on, we'll see that what are the minor differences between the enzymes and the catalyst. Okay. So these enzymes, they have extraordinary catalytic power, okay, often far greater than of the ordinary synthetic catalysts. Okay, so the enzymes, they have a extraordinary catalytic power. Say, for example, that the reaction between water molecule and the carbon dioxide. Okay? So the water, it combines with the carbon dioxide to form the carbonic acid. So this reaction, in absence of any enzyme, it is very less. Okay, it is very slow. But when the enzyme acts on this reaction, say there is in our body system, there is one specific enzyme which is called as the carbonic anhydrase. Okay, carbonic anhydrase. So this particular enzyme, it acts on to increase this rate of the reaction. That is the combination of water and the carbon dioxide to form the carbonic acid. So when this particular enzyme acts on this, the rate of the reaction it tremendously increased. Okay? It is 10 to the power 20 times. It is increased. So the rate of the reaction, it is increased like anything when the reaction is acted upon by the enzymes. Okay. So that means the enzymes, they have the extraordinary catalytic power. Okay. Then these enzymes, they are highly specific in their action and accelerate specific chemical reactions. So that means these enzymes, they are specific for a particular type of the reaction. Say the enzyme carbonic anhydrase. So it is specific. This will act only on the combination of the water and the carbon dioxide to form the carbonic acid. This particular enzyme will not act on the conversion of the glucose to, say, some other product, okay, or say hydrolysis of the protein to the amino acids, or the degradation of the triacylglycerols to glycerol and fatty acid. This enzyme is not going to act. This enzyme will act only in the reaction of the water molecule combining with the carbon dioxide to form the carbonic acid. That means this enzyme is very specific for that particular reaction in which the water molecule combines with the carbon dioxide to form the carbonic acid. This is not going to act in the other reactions. So that means this particular enzyme is very specific for the type of the reaction or the specific reaction that it catalyzes. Okay. Next is that the enzymes function in the aqueous solutions under very mild conditions of the temperature and the pH. So there is a specific range of the pH in which the enzymes act, or there is a specific range of the temperature in which the enzymes will be active. They will be performing the function. Okay? So beyond that, the enzymes will not be functioning. They cannot express their activity. It means they have a limited range of the temperature and the limited range of the pH in which they express their activity. Okay. Now let's see briefly the history of the enzymes. Okay, because history of the enzyme is important because the history of the biochemistry itself is the history of the enzyme. So you can say that history of the enzyme is the history of the biochemistry. So much of the history of the biochemistry is the history of enzyme research. Okay. So enzymes were first recognized and described in the early 1800 in the studies of the digestion of meat by the secretion of the stomach and the conversion of the stomach into the conversion of the starch into sugar by saliva and various plant extracts. So how the interest of the enzymes develops? It develops with that the people started asking them how the meat is digested by the stomach secretion. Okay, or how the starch is digested or is converted to the sugar, okay, by the saliva or by some of the plant extracts. So people started asking this question, that how these complex molecules, they are digested to the simpler molecules. So okay, so with this, that thinking that how it has come and all that, so it starts this one discussing about the enzymes. In 1836, Berzelius coined the term catalyst. Okay? So the term catalyst, it was coined in 1836 by Berzelius, and the meaning of catalyst means it is to dissolve. Okay? Catalysis means it is to dissolve. In 1860, Louis Pasteur, he concluded that fermentation of the sugar into alcohol by the yeast is catalyzed by the ferment. So Louis Pasteur, he thinks that the conversion of the sugar to the alcohol, or we call it as the formation of the alcohol from the sugar, it is done by certain elements or certain compounds, which he called it as the ferments. Okay, so he postulated that these ferments are inseparable from the structure of the living cells. Okay, and he feels that these ferments, or that material which is converting the sugar to the alcohol, it should be present in the cell itself. Okay, it should be present in the cell system. That is, that time the formation of the alcohol from the sugar, it was done by using the yeast cell. So he feels that these molecules, which is converting the sugar to the alcohol, it should be present in the yeast cell. Only then they can catalyze, they can add or convert the sugar to the alcohol. That was his hypothesis. Okay. In 1878, Kuhnke used the what enzyme. Okay. So this one you have to remember that who coined the term enzyme. Okay. So the enzyme was for the first time used by Kuhnke in 1878. So he means that enzymes means in the yeast. Okay. So it is just to indicate that catalysis taking place in the biological system. So Kuhnke, he used the word enzyme to indicate that the catalysis of the reaction that is being catalyzed by the catalyst, it should occur in the living cells. In 1883, the term substrate was introduced by Doclocks. Okay? So in biochemistry, that reactants, we call it as the substrate. Okay? So in chemistry, the reactants, okay, we call the groups or the compounds which are reacting to form the product, we call it as the reactants. But in case of the biochemistry, we call it as the substrate. So substrate means it is the reactants. Okay? In 1897, Edward Buckner discovered the yeast extracts can ferment the sugars to the alcohol. Okay, so earlier Louis Pasteur, he postulated that this ferments has to be present in the cells. That means the fermentation process, that conversion of the sugar to the alcohol, it can occur only in the living cell. That was the hypothesis of the Louis Pasteur. Okay, but in 1897, the Edward Buckner, he was performing some experiment, and he found that the fermentation, it can occur outside the living cells. So that means when the yeast cells, they are broken, if the soluble fraction is extracted from the cells, okay, the liquid portion from the cell, it is removed from the cells, still that extract, it can cause the fermentation of the sugar. That means the fermentation is not required to be present in the cell. Okay? Louis Pasteur said that it has to occur in the cell, but Edward Buckner, he found that no, it is not that that the cell is required for the fermentation, but the content of the cell which is required for the fermentation. Okay, so that proved that the enzymes involved in the fermentation can function when removed from the structure of the living cells. So this fermentation, it doesn't have to be a process that occurs in the living system. In 1926, James Sumner isolates and crystallized the urease. Okay? So he isolated one enzyme which is called as the urease from the jet bean extract. Okay, so the jet bean was used. From there, he isolated and crystallized the urease. Okay, and so this provides a breakthrough in the early studies of the specific enzymes, and he found that the urease crystal consisted entirely of the protein and postulated that all the enzymes are a protein. So what he did, he isolated the urease enzyme from the jet bean, then it was crystallized. Okay? Then he studied the chemical nature of these enzymes. Okay? So in that, what he found was that the enzyme is entirely made up of amino acid. It is a protein. Okay? So he postulated that the enzymes are the proteins which has the catalytic activity. In 1930s, John Northrop and Moses Kunis crystallized the enzymes pepsin and trypsin, and they also found that this trypsin and pepsin is also a protein. Okay, so similar finding with that of the James Sumner. James Sumner, he has postulated that the enzymes are protein. Then the John Northrop and Moses Kunis, they also crystallized the two enzymes, that is pepsin and trypsin, and they also found that the chemical nature of these enzymes, it is the protein in nature. So this removes the controversy of the Sumner's proposal that enzymes are protein. So there was some controversy when the James Sumner proposed that the proteins are the enzymes are the protein. So again in the subsequent finding, the John Northrop and the Kunis, they also found that the two enzymes, they are of the protein, or the chemical nature, chemical composition is the protein. So that removes that controversy. In 1930s, JBS Halden wrote Enzymes. Okay? So here it contains the remarkable suggestion that weak bonding interaction between the enzymes and its substrate might be used to distort the substrate and catalyze the enzymatic catalysis. So Halden, in his book Enzymes, what he suggested that now there may be some sort of the weak interaction between the enzyme molecule and the substrate molecule. So because of this weak interaction between the enzyme molecule and the substrate molecule, the structure of the substrate molecule may be distorted. Okay? It may not remain in the native conformation. And this distortion in the structure of the substrate molecule, it helps in the catalysis. Okay, so that was the suggestion, and this was a very remarkable suggestion, because later on we may come across that many of the reactions in which the enzymes catalyzes, it distorts the conformation of the substrate molecule. Okay, so what he has suggested, it is a clue that comes through. In 1946, James Sumner was awarded Nobel Prize for his credit of isolation and crystallization of the urease from the jet bean extract. Okay? Because James Sumner, he was the first one to isolate the enzymes and crystallize it. Okay? But he has done it in 1926. That is the isolation and the crystallization of the urease. Okay? So since that was the pioneer work, that was recognized, and he was awarded a Nobel Prize in 1946. Okay, then why there is a need to study the enzymes? Why we need to know about the enzymes? Okay, so what is the importance for the study of the enzymes? The study of the enzymes has the immense practical importance. Like say, in some diseases, especially the inheritable genetic disorders, there may be deficiency or even total absence of one or more enzymes in the tissues. Okay, so some of the genetic disorders, say you must have heard about the albinism. Okay? So in case of the albinism, that is a disease condition in which the melanin is not synthesized. Okay? So melanin, it is a pigment synthesized from the amino acid tyrosine. Okay? So when the amino acid tyrosine, it cannot be metabolized to form the melanin because of the absence of one enzyme, then it causes the albinism. Okay, so what is this disease? this disease is caused due to the absence of a particular enzyme, due to the deficiency of a particular enzyme in our system. Okay, so that means when some enzymes is deficient, when it is lacking, when they are not produced in the body, it may cause the diseases. Okay, so that means if you want to know the disease condition, then you need to know about the enzyme. Okay, so this is one importance for the study of the enzyme. The second is that abnormal condition can also be caused by the excessive activity of specific enzymes. So some specific enzymes, it may be produced in the excessive condition. Okay? They may be produced in the excess amount. So when it is produced in the excess amount, that also may lead to some disease conditions. Okay? Measurement of the activity of certain enzymes in the blood plasma, erythrocytes, or the tissue samples are important in diagnosing diseases. Okay? So in the diagnostic biochemistry, many of the enzymes which are present in the blood, okay, we used to measure it. Okay? So when there is some disease condition, the level of these enzymes in the blood increases. So by checking the level of that particular enzyme in the blood, we can get a clue that there may be problem in these disease conditions. Okay, say alkaline phosphatase, it may be increase in the liver diseases. Okay, say then in case of the heart attack, okay, myocardial infarction, then 3 enzymes, they are elevated in the blood. Like say one is the SGOT, okay, serum glutamate oxaloacetate transaminases, then lactate dehydrogenase, then creatine phosphokinase. So these 3 enzymes are increased in level in the blood in case of the heart attack. Okay? So if we suspect that somebody is having the heart attack, we can check the level of these enzymes in the blood. From there we can get some clue. Okay? Or say there may be a disease condition in the liver. So in liver diseases, many enzymes are elevated, say like one the alkaline phosphatase, it may be the LDAs, it may be the SGPT. Okay, so like that, many enzymes are elevated in the case of the liver diseases. Okay? Say if you have the prostate cancer, okay, in case of prostate cancers, then the enzyme acid phosphatase is increases. So like that, in different disease conditions, the level of the certain enzymes in the blood or in the tissues is increased. So by checking the level of these enzymes in the tissues or in the blood, we can get some clue that there may be some problem with this organ or the tissues. Okay. Next is that enzymes have also become important tools in the medicine, okay, in the chemical industry, food processing industry, and the agriculture. So enzymes, it has the wide application in the industry, agriculture, and all that. Now you must have heard of the detergents containing the enzymes. Okay, then you have the meat processing industry in which the enzymes are used. So like that, the enzymes, it has a very wide application in the industry, in agriculture, in medicine, and all that. So again, until and unless we know fully about the enzymes, we cannot apply the enzymes in these purposes. Okay, so we need to study the enzymes. Enzymes also plays important roles in the everyday activities in home, such as the food preparation, cleaning, etc. Okay, so food preparation, you must have heard about the papain, the enzyme papain, which is extracted from the papaya. So this is used for the digestion of the meat, okay, meat products and all that, or the softening of the meat and all that. So like that, enzymes, it has a very wide application, and we need to know the importance of the enzymes. That is the reason that we have to study the enzymes. Okay, how does the enzyme differ from the catalyst? Okay, so catalysts, they also catalyze the reaction, they also increase the rate of the reaction. Same thing, the enzymes also increase the rate of the reaction. So, but there is some differences between the enzymes and the catalysts. Okay, so what are the differences? First, the enzymes are organic in nature. They are either protein or ribonucleic acids. So this, it is not recent now. I think around 1980, the ribosomes were discovered. Okay. So before that, before 1980, Before 1980, it was known that all the enzymes are the protein in nature, but in 1990, one scientist called Thomas Jake, he discovered ribonucleic acid. So now we know that this one, some of the RNAs, ribonucleic acids, they also has the catalytic activity. So except this ribozymes, that okay, so that RNAs which has the catalytic activity, we call it as the ribozymes. So except for the ribozymes, the other enzymes are all proteins. Okay? While the catalysts are inorganic in nature. So the first difference between the enzymes and the catalysts is that the enzymes, they are organic in nature. It is mainly the proteins, or some of the enzymes are ribonucleic acid, while the catalysts are inorganic in nature, like say nickel, you have then lead and all that. Okay, so most of them, they are inorganic in nature. The second point is that the enzymes are highly specific in their action, but the catalysts are not as specific as that of the enzyme. So the enzymes, they will be catalyzing a specific reaction, or a very closely related reactions, they may catalyze it. But the catalysts, they catalyze a wide range of the reactions. They are not specific for a particular reaction, but it may catalyze a wide range of the reactions. Third is that the enzymes have extraordinary catalytic power. I have already given the example of the carbonic anhydrase. Okay, so this extraordinary this catalytic power, it is often far greater than of the synthetic or inorganic catalyst. Okay, the synthetic or inorganic catalyst, they may not have that much high catalytic activity compared to that of the enzymes. Then the fourth one is that enzymes function in aqueous solutions under very mild conditions of the temperature and pH. But the catalyst, it may function in a very wide range of the pH or a very wide range of the temperature. Okay, so these are some of the basic differences between the enzymes and the catalysts. I think we'll stop here. Okay. Next time we'll discuss about the chemical nature of the enzymes. So for today we'll stop here. It's just I have given you the introduction to the enzymes. So let's stop with this. Or if you have any questions, acids. Okay. So proteins, we know that they are made up of the amino acid, whereas the RNA, it is made up of the ribonucleotides. So all these are organic in nature. So all the enzymes, they are proteins with the exception of a small group of the catalytic RNAs. So except for this small group of the catalytic RNAs, the other enzymes are all proteins. And since these are enzymes are the proteins or the RNAs, then they have to have the native conformation to express their activity. Okay, so that means their catalytic activity depends upon the integrity of their native conformation. If the native conformation of these molecules is distorted or it is disturbed, then they will not be able to express their activity. And we know that what is their activity? Their activity is to catalyze the reaction. So that means these enzymes, whether it is the protein or the RNA, if their native conformation is destroyed, then they cannot catalyze the reaction. They will not have the catalytic activity. Okay. So what is the native conformation? We know that this in case of the proteins or the nucleic acids, the native conformation is maintained by many forces. Okay, so that is the secondary, tertiary, quaternary structure of these proteins. It has to be maintained. Then, if the protein contains more than one subunit, then the quaternary structure, it has to be maintained. That is, the addition of the subunits in the protein molecule has to be in the native conformation. So until and unless these molecules are in the native conformation, they will not be able to perform their function. That is, they will not have the catalytic activity. Okay, like the other proteins, the enzymes, they have the molecular weights ranging from about 12,000 to over 1 million Dalton. Okay, because they are also the proteins, so their molecular weight or the size will also be same as that of the other proteins. Okay, then some of the enzymes, it does not require any additional group. Okay, they do not need any additional group to catalyze their catalyze the type of the reaction or to express their activity. But there are some enzymes which require some additional groups in addition to the amino acids to perform their function. So these additional groups which are required by the enzymes to express their activity are called as the cofactor. So what is cofactor? So cofactor is the additional group required by the enzymes to perform their function. Okay, so that is the cofactor. Now these cofactors, it may be either inorganic ions or it may be complex organo or metallo-organic molecules. Okay, so these coenzymes, cofactors, it may be inorganic ions like say iron, magnesium, zinc, copper, and so on. Okay, so they may be only the inorganic ions, or it may be some organic molecules like say thiamine pyrophosphate, then the nicotinamide adenine dinucleotide, flavin adenine dinucleotide, pyridoxal phosphate. So all there are so many. We'll come to that when we discuss the vitamins or the metabolism part. Okay, so it may be inorganic, it may be organic molecules, or it may be a combination of the inorganic as well as the organic molecules. So that is the metallo-organic molecules. Okay, so all this, the coenzyme, it includes the inorganic ions, it includes the organic complex organic molecules, or it is a complex of the metal ions and the organic molecules. So that all is collectively called as the coenzymes. Now some enzymes require both the coenzyme and one or more metal ions for activity. So some of the enzymes, it may require only the metal ions, some may be require the organic molecules, some may be requiring more than one or this one cofactors. Okay, so it is not that only one may be required. The enzyme may require more than one additional components. Okay, now here what we have defined is the cofactor. Okay, so cofactor, it includes the inorganic as well as the organic molecules. But when it is purely organic, okay, purely organic, then we call it as the coenzyme. So what is the difference between the cofactor and a coenzyme? Cofactor, it includes the metal ions and the organic molecules, whereas the coenzymes, it includes the organic molecules. Okay, and most of these are the derivatives of the vitamin, mainly the water soluble vitamins. That there are some fat soluble vitamins like the vitamin K. Okay, but most of the coenzymes, they are of the derivatives of the vitamin B complex. Okay. Now, here I'm giving you some of the metal ions which are required for this enzyme. So that means these are the cofactors. Okay, some inorganic elements that serve as the cofactors for the enzymes, like say copper. Okay, the copper ion, it is required for the cytochrome oxidase. So the cytochrome oxidase, to express their activity, to catalyze the reaction, they need the copper ion. Okay, iron in the ferric or the ferrous state, it is required by the cytochrome oxidase, catalase, peroxidase. Okay, then the potassium, it is required for the pyruvate kinase. Magnesium, it is required for hexokinase, glucose 6-phosphatase. Manganese, it is required for the arginase, ribonucleotide reductase. Molybdenum, it is required for the dinitrogenase. Nickel, it is required for the urease. Selenium, it is required for glutathione peroxidase. Okay. Then zinc, it is required for the carbonic anhydrase and the alcohol dehydrogenase. Okay, so here there are many enzymes which requires the inorganic metal ions, which we call it as the cofactors. Okay, now this complete catalytically active enzyme. So that means it is the protein component as well as the non-protein component or the cofactor. So this is called as the holoenzyme. Okay, so a complete catalytically active enzyme together with is the coenzyme and all the metal ions is called as a holoenzyme. So holoenzyme, that means it includes the protein part, that is the enzyme. Then it also includes the non-protein part, that may be the organic molecules, which some of them which I have mentioned it, or the metal ions. Okay, so that is called as the holoenzyme. The protein part of such enzyme is called as the apoenzyme or the apoprotein. So apoenzyme or the apoprotein means it is the protein component of the holoenzyme. Okay. The coenzymes function as transient carrier of specific functional group. So this is the function of the coenzymes. What is the function? The coenzyme it acts as a carrier. Okay. The transient carrier of the groups during the course of the reaction. Okay. So holoenzyme, this is the active enzyme component, is equal to apoenzyme plus the coenzyme. Okay, so that means holoenzyme minus the apoenzyme, it becomes the coenzyme, or you can say the holoenzyme minus the coenzyme, it is the apoenzyme. Okay, the term prosthetic group is used when the non-protein moiety, non-protein part is tightly bound to the apoenzymes. Okay, so if that the metal ion or the organic group, it is tightly bound, means it is covalently attached to the enzyme component, then it is called as the prosthetic group. Remember this one. Okay, remember these terms, that is what is the cofactor, what is coenzyme, and what is prosthetic group. Okay, the coenzyme can be separated by dialysis from the enzyme, while the prosthetic group cannot be separated since they are covalently attached. When these prosthetic groups are covalently attached to the enzyme, then it may not be possible to remove it easily. So they cannot be removed by the process of the dialysis. Only they can be separated from the enzyme only when the bond which links these molecules to the enzyme is broken. Okay, so that means this cannot be separated by the process of the dialysis. Okay. One example is that you'll come across when we discuss the TCA cycle, okay, tricarboxylic acid cycle. There is one enzyme which is called as the succinate dehydrogenase. Now, this succinate dehydrogenase requires flavinin adenine dinucleotide. It requires the FAD. Okay, so this FAD is covalently attached to the active site of the enzyme. So since they are actively attached to the coenzyme, they cannot be removed from the enzyme by the dialysis. So this acts as a prosthetic group. Okay, so prosthetic group means it is the cofactor or the coenzyme which is tightly bound or covalently attached to the enzyme molecule. Okay, the word monomeric enzyme is used if the enzyme is made up of a single polypeptide. So if the enzyme has only one polypeptide chain, only one peptide on the polypeptide chain, then that particular enzyme is called as a monomeric enzyme. Okay, the examples are ribonuclease and the trypsin. So ribonuclease and trypsin, they have only one polypeptide, so they are called as the monomeric enzyme. But there will be many enzymes which contains more than one peptide or the polypeptide chain. So what they are called as the oligomeric enzymes. Okay, some enzymes possesses more than one polypeptide or the subunit chain, and they are known as the oligomeric enzymes. The examples are the lactate dehydrogenase. So lactate dehydrogenase, we know that they have 4 subunits. Okay, then aspartate transcarbamylase, etc. So there are many enzymes which has more than one subunit, more than one polypeptide chain or the peptide chain. So if that particular enzyme, if that has more than one subunit, then we call it as the oligomeric enzyme. So what we are learning here, we are learning that the enzymes, they may be monomeric enzymes, means it contains only one polypeptide or the peptide chain. The second one we call it as the oligomeric enzymes if they contain more than one polypeptide or the peptide chain. Okay, there are certain multi-enzyme complexes. Okay, so there are some enzymes which have more than one enzyme. Okay, they have means these enzymes it can perform more than one function. It can catalyze more than one type of the reaction. So it is just because that complex it contains more than one active site. They have more than one enzyme. Okay, so 2 or 3 or more enzymes it comes together to form a protein molecule. So since 2-3 enzymes are coming together to form a complex, they will be able to catalyze more than one type of the reaction. So when more than one enzyme molecule it comes up together to form a complex, then we call it as the multi-enzyme complexes. Okay, so there are certain multi-enzyme complexes possessing specific sites to catalyze the different reactions in a sequence. Okay? Only the native intake multi-enzyme complex is functionally active and not the individual units. So here, when they come together, that complex only will be active. When they are separated, then they may not have the catalytic activity. If they are separated, for example, the pyruvate dehydrogenase. Okay, so pyruvate dehydrogenase it has 3 enzyme subunits. Okay, they have the 3 catalytic sites. Then another one is the fatty acid synthesis, then prostaglandin synthesis, etc. So all these enzymes they exhibit the activity only when the different enzymes they are in the complex form. Okay, the enzymes exhibit all the general properties of the protein. So what we have learned here, what is that we have learned that the enzymes they may be of the monomeric, this one enzymes. Okay, that is what we said, the monomeric enzymes. Then some of them may be oligomeric enzymes. So that means it contains more than one peptide or the polypeptide chain, but they have only one catalytic activity. They will catalyze only one type of the reaction. Then the third one is the multi-enzyme complexes. So here, more than one enzyme come together to form a complex. So since this complex is having more than one enzyme, they will be catalyzing more than one type of the reactions. Okay, so all these things will be coming across when we start the metabolism part. Okay, then the next question is how the enzymes work. Okay, how these enzymes it works. That means how these enzymes it increases the rate of the reaction. The function of the enzyme we know that is that it increases the rate of the reaction without itself being consumed. Okay, so the question is that how this enzyme it increases the rate of the reaction. So enzymes catalyzed reaction might be written as, okay, so this is the general reaction for the enzyme catalyzed reaction. That enzymes here, E stands for the enzyme, S for the substrate, then P for the product. Okay, so enzymes it combines with the substrate to form the enzyme substrate complex. Now this enzyme substrate complex is converted to the enzyme product complex. Then finally the enzyme product complex is dissociated to form enzyme and the product. So here what we are seeing here, that the enzyme was initially consumed to form the enzyme substrate complex. Then at the end of the reaction, the enzyme itself is released out. So that means the enzyme itself is not consumed at the end of the reaction. Okay, so enzymes increases the rate of the reaction without affecting the equilibrium. So here it shows the equilibrium. Okay, so this enzymes it does not affect the equilibrium, but simply that what the thermodynamically feasible reaction it increases the rate. It doesn't change the equilibrium. Okay, so in its normal state, so this is just a before we start about the enzyme in details, we need to have a background. Okay, so that in its normal state form, in its normal stable form or the ground state, any molecule such as the S and P in this reaction. Okay, in this graph now. Here we are showing one as the S, that is the ground state, then another one is the P, that is also in the ground state. The S stands for the substrate, then P it stands for the product. So that substrate and the product, they are having certain amount of energy level. Okay, so in this one, that the energy level of the S is higher than the P. So this is the above line, it is the energy level of the substrate. Then lower one is the energy level of the product. So that means there is a difference in the energy level between the substrate and the product. So the substrate will have the tendency to form the product because the energy level of the product is lower than that of the substrate. So this substrate, it will tend to convert to the P. Okay, so that is thermodynamically feasible. Why it is feasible? Because there is a release of the energy. The energy level of the compound that is going to form after the reaction, it is lower. Okay, so since it is lower, then that reaction will be favorable. It is thermodynamically feasible. Okay, so just see whether that reaction will be feasible or not. You can just know it by knowing the energy level of the compound. So if the energy level of the substrate is higher than that of the product, then the reaction is feasible. Okay, now, so the reaction will occur. The reaction, that is the conversion of this S to the P, will naturally occur without the enzymes also. But the rate at which this substrate is converted to the P will be very slow in absence of the catalyst or the enzyme. So what does the enzyme do? The enzyme, it increases the rate for conversion of the S to the P. Okay, so that is the role of the enzyme. It is just to increase the rate of the reaction. Now here, the equilibrium between S and P reflects the differences in the free energy of their ground states. For example, here the free energy of P, that is the product, is lower than that of the S. So equilibrium favors the conversion to the P, that is formation of the P. There is a energetic barrier between S and P that represents the energy required to alignment of the reacting groups. Formation of transient unstable charges, bond rearrangements, and the other transformation for the reaction to occur in the direction. So here, what happened is that when this S has to be substrate, S has to be converted to the P, then it has to attain a certain amount of the energy level. Okay, so that it has to attain this transition state. Okay, so what is the transition state? This is the energy barrier. Okay, so here this is for the substrate, the energy of this substrate. Then you have the transition state. This is the energy level. So energy level of the transition state is higher than that of the substrate. So this substrate, it has to pass through this transition state. Only then this will be converted to the P, to the product. So that means this substrate, it has to attain this energy, then it has to come to the P. So this is an energy barrier. Okay? So this energy barrier, why it is required? So this represents the energy required for the alignment of the reacting group. So in the substrate itself, there will be groups which are aligning to form the product. Okay, to the reaction to occur. Okay, then this is a the bond rearrangement, because some bonds it has to be formed, some bonds has to be broken. So there should be some rearrangement of the bonds in the compound. Okay, then other transformations. So all these things, the rearrangement of the aligning of the reacting groups, then the bond rearrangements, then other transformations which are required for conversion of the S to the P, it is happening in this energy barrier. Okay, so that is the energy barrier. So that means this S substrate has to attain the transition state, only then this will be converted to the P. So the role of the enzyme here is that it decreases. Just see the lower graph. So here, when the substrate, it combines with the enzyme, it forms the enzyme substrate complex, then enzyme product complex, then the P. So here this is the energy level of the enzyme substrate complex and energy enzyme product level. So this is the level of that complex, and the above one, this line, the above line, it is the energy level of the transition state. So what the enzyme is doing, that it is decreasing. Okay, it is decreasing the transition state, the energy level of the transition state. So the function of the enzyme is to decrease the energy level of the transition state, so that it is easily, the substrate is easily converted to the product. Okay, so that is how the enzymes are enhancing the rate of the reaction. Yes, any question in this part? Yes, anyone? You have any question? Yes, anyone? Is there any question on this part, what I'm discussing? No, no one. Okay, we'll continue then. Okay, so now we know that how the enzymes, it enhances the rate of the reaction. So enzymes enhances the rate of the reaction by decreasing the energy transition, the energy level of the transition state. Okay, so transition state is not a chemical species with any significant stability. It is simply a fleeting molecular moment in which the events such as bond formation and the charge development have proceeded to the precise point at which a collapse to either substrate or the product is equally likely. So this, the transition state, it may have the equal opportunity to convert it to the product or to the substrate. Okay, the difference between the energy levels of the ground state and the transition state is called as the activation energy. So what is activation energy? So this is just the difference in the energy level of the substrate in the ground state and the energy level of the at the transition state. Okay, the rate of the reaction reflects this activation energy. Enzymes enhances the rate of the reaction by lowering the activation energy. So the role of the enzyme is just to decrease the energy level. Okay, the activation energy. Okay, then how the enzyme substrate complex are formed and whether it is required. Okay, the formation of the enzyme substrate complex is required for the catalysis of the reaction. Okay, so the prime requisite for the enzyme catalysis is that the substrate must combine with the enzyme. Okay, so what is the requirement for the catalysis of the reaction? The requirement is that the enzyme and the substrate, it should combine to form the enzyme substrate complex. So this is a prerequisite. It is a required. Okay, it is the requisite step, which ultimately result in the product formation. So if there is no enzyme substrate complex formation, the result may not be formed. So the required step is that the enzyme should combine with the substrate to form the enzyme substrate complex. Okay. The molecule that is bound by the active site and acted upon by the enzyme is called as the substrate. So what is the substrate? Substrate in chemistry we call as the reactant. So this is the molecule that has to be bound by the enzyme and acted by this enzyme. The binding of the substrate to the enzyme occurs at a particular site, okay, on the enzyme molecule, which is called as the active site of the enzyme. So the substrate will not bind to any, it cannot bind to any part of the enzyme. It has to bind to a specific location on the enzyme molecule, and that specific location on the enzyme molecule where the substrate binds is called as the active site. Okay. The idea of the enzyme substrate complex was first conceived by Michaelis and Mendel in 1913, which is accepted even today by the modern biochemists. So it was already revealed in 1913, and since it is true, it is still true. Okay. So there are some evidences which suggest the formation of the enzyme substrate complex. So in the previous slide, I have mentioned that for any enzyme to catalyze the reaction, the formation of the enzyme substrate complex is required. Okay. So since we are studying science, we should have some evidence to show that what we propose it is correct. That is, there should be some evidences to show that the enzyme combined with the substrate to form the enzyme substrate complexes. Okay. The first evidence is that at a constant concentration of the enzyme, just let me go through what is there, then I'll explain it with the graph. Okay. So at a constant concentration of the enzyme, the reaction rate increases with the increasing substrate concentration until a maximum velocity is reached. Okay. In contrast, uncatalyzed reactions do not show this saturation effect. In 1913, Leonard Michaelis interpreted the maximum velocity of an enzyme catalyzed reaction in terms of the formation of a discrete ES complex. Okay. So at sufficiently high substrate concentration, the catalytic sites are filled, so the reaction rate reaches a maximum. So this is indirect, but this is the most general evidence for the formation of the enzyme substrate complex. So what it says is that in this graph, if you see it, then see in the x-axis you have the substrate concentration. Okay, so you are just increasing the substrate concentration. On y-axis you have the velocity of the enzyme catalyzed reaction. So initially, when the concentration of the substrate is low, or we call it at zero, then velocity of the reaction, that is the rate at which the substrate is converted to a product, is also zero. Okay, so at this point the substrate concentration is zero, then rate of the reaction is also zero. But with increasing substrate concentration, this rate of the reaction, it is increasing. Okay, it is increasing, then it forms a hyperbolic graph. Then at a certain point, okay, at this level, which is the maximum velocity, the increase in the substrate concentration, it doesn't have any effect. Okay, so the increase in the substrate concentration is not going to increase in the velocity of the reaction. This is unique for the enzyme catalyzed reaction, unlike that of the catalyst. In the case of the catalyst, you will find a straight line. Okay, at the zero substrate or the reactant, the reaction will be low. Then with the increase in the substrate concentration, it also increases. It forms a straight line. Okay, but in case of the enzymes, you will not find any straight line, but you will get a graph like this. Okay, so after some point, the increase in the substrate concentration, it does not reflect in the increase of the enzyme concentration. Now this is just because that at this point, okay, at this point where the maximum velocity has reached, all the enzyme molecules are saturated with the substrate. So further increase in the substrate concentration will not lead to the formation of the more enzyme substrate complex. So the rate of the reaction remains the constant at this point. Okay. The second evidence for formation of the enzyme substrate complex, it comes from the spectroscopic characteristics. Okay. So many enzymes, they contain the groups which we call it as the prosthetic group. Okay, so the spectroscopic characteristic of many enzymes and the substrates change upon formation of the enzyme substrate complex, and these changes are striking if the enzyme contains a color prosthetic group. Okay, for example, the tryptophan synthase. So this is a bacterial enzyme that contains pyridoxal phosphate as the prosthetic group. Means the pyridoxal phosphate is covalently attached to the enzyme molecule. Okay, so this particular enzyme, that the tryptophan synthase, it illustrated the spectroscopic characteristics. Okay, so this enzyme, it catalyzes the synthesis of the L-tryptophan, that is one amino acid, L-tryptophan from the L-serine and the indole. So here the substrate is L-serine and the indole, and that is combined to form the L-tryptophan. So The addition of the L-serine to the enzyme produces a marked increase in the fluorescence of the pyridoxal phosphate group. So here you just see in the graph. Okay, so here the middle one, which is shown in the black color, it is enzyme alone. When you take the enzyme alone, you take the spectroscopic characteristics. Okay, just you check it the fluorescence. Then this is the graph that we are getting when the enzyme is alone, when there is no substrate is added. Now to this solution which contains the enzyme, when we add the first substrate is the L-serine. So when the L-serine is added, then its fluorescence is increased like anything. So here the one which is shown in the red color. So just you can see how much it is increased. Okay, so from the black, from the middle one, the red one, above one. Okay, so it is drastically increased. So why is it so? It is just because that the enzyme is linking with the L-serine, the first substrate. It forms a complex with the enzyme, so because of that there is increase in the fluorescence. Okay, then in the next step, when the second substrate is added, so the second substrate is the indole. When it is indole, then this indole is added, then the fluorescence is drastically reduced. It becomes to the lower one, which is shown in the blue color. Okay, so this blue color is even lower than that of the enzyme alone. So that means the addition of the second substrate indole, it quenches, okay? It reduces the fluorescence. So all this increase in the fluorescence with increase in the with the addition of this serine, and the decrease in the fluorescence with the addition of the indole, it is all because there is a complex formation between these substrates and the enzyme. Okay, so this gives a proof that there is a formation of the enzyme substrate complex when the enzymes catalyze the reaction. Okay, the third evidence, it comes from the electron micrography. Okay, so enzyme substrate complexes have been directly visualized by electron microscopy. Okay, as in the electron micrograph of the DNA polymerase 1 bound to the DNA template. So DNA polymerase 1 is an enzyme which is involved in the DNA synthesis. Okay, so that complex of this particular enzyme with the DNA, it has been visualized by electron microscopy. Okay, so that means the enzyme and the substrate complex is formed. Okay, so in addition, the complex of the enzymes with the suitable with the substrate or its analogs have also been isolated in the stable form. So many enzymes with the substrate bound together, okay, or the substrate covalently attached to the enzyme molecule has been visualized or has been identified. Okay, so these are the evidences which suggest that there is a formation of the complex of the enzyme and a substrate that is called as the enzyme substrate complex, and this is a required step for the enzyme catalyzed reactions. Okay, now coming to the classification of the enzymes. So enzymes are generally named after adding the suffix "ase" to the name of the substrate. So what happens generally when the enzymes are classified, or when the name of the enzyme is given, then the name "ase" is added to the substrate. Okay, so like the enzymes acting on the nucleic acids are known as the nucleases. So we're adding the "ase" here, nucleases. That it comes from the nucleic acids. Okay, enzymes hydrolyzing the dipeptides are known as dipeptidases. Okay, dipeptidases. Then dipeptidase should be some suppression should be there. Dipeptidase. Okay, then urases catalyzes the hydrolysis of the urea. So the urea is hydrolyzed, substrate is the urea, so the name of the enzyme is called as the urases. DNA polymerases catalyze the polymerization of the nucleotides to form the DNA. So that polymerase means it is indicating that it is the polymerization. So what polymerization is going on? that polymerization of the monomer units to form the DNA. So it is called as the DNA polymerization. Okay. The enzymes were sometimes named by discoverers for a broad function. Okay. So some enzymes, it may have the broad activity, so the broad name will be given. Before the specific reaction catalyst was known. Okay. Say an enzyme known to act in the digestion of the fruits was named as pepsin, from the Greek word pepsis, which means digestion. Okay, so the word pepsin, it is coming from the Greek, which means that it is digestion. So that time, what type of reaction is occurring, that was not known, but it knows that this particular enzyme is required for the digestion. Okay, digestion of the food material. So it is called as the pepsin. Okay. So then the lysozyme was named for its ability to lyse bacterial cell. So since it has the ability to lyse the bacterial cell, they are called as the lysozyme. Okay. Still others were named for their source. Okay, like say trypsin, named in part from the Greek train, to wear down. Okay, was obtained by rubbing the pancreatic tissue with the glycerin. Okay. Sometimes the same enzyme has two or more names. Okay, so one enzyme, it may be having two or more names, or the two different enzymes have the same name. Okay, say the enzymes which is the present in the saliva. So we know that it is the ptyalin. We also call it as the amylase. Okay, so it is the same enzyme, but it is having the different names. So like that, some enzymes they may have the two or more names, or sometimes it is also possible that for the same name is given to two or more enzymes. So there is some ambiguities. Okay, so because of such ambiguities and the ever increasing number of newly discovered enzymes, biochemists, by international agreement, adopted a system for naming and classifying enzymes. So earlier we mentioned that there are some ambiguities in naming the enzyme. So to remove these ambiguities, the biochemists all over the world, they agreed, okay, to have a systematic naming of the enzymes. Now this system, it divides the enzymes into 6 classes. So all the enzymes which are present, it is divided into 6 groups or the 6 classes, each with the subclasses based on the type of the reaction catalyst. Okay, so each enzyme is assigned a 4-part classification. So 4-digit classification is given for each enzyme, and a systematic name is also given. So what is given? That a 4-digit classification of the enzymes and also a systematic name of the enzymes is given for individual enzymes. So which identifies the reaction it catalyzes. Okay. Say example, the formal systematic name of the enzyme catalyzing the reaction, that is ATP combines with the D-glucose to form ADP and the D-glucose 6-phosphate. So here what happened? One phosphate group from ATP is transferred to the D-glucose to form the D-glucose 6-phosphate. Okay. So now this is ATP glucose phosphotransferase. So what type of reaction is happening? It is the transfer of the phosphate group from ATP to glucose. So it is ATP glucose. Then what is transferred? The phosphate, the phospho. Then type of the reaction is the transfer of the group. So it is called as the phosphotransferase. So this is the systematic name. Okay. So this indicates that it catalyzes the transfer of a phosphoryl group from ATP to glucose. And its enzyme commission number, that the 4-digit enzyme classification number, is 2.7.1.1. So there are 4 digits are assigned to this particular enzyme. So this 2.7.1.1 is the specific number given to this particular enzyme, that is the ATP glucose phosphotransferase. Okay, the first number, that is 2 here, it denotes the class name, that is the transferase. That this group of the enzyme, it catalyzes the transfer of a group, that is the transferase.
The second group, the second number, that is 7, the subclass phosphotransferase. So what group is transferred? It is the phosphate. So that if the group transfer is phosphate, the number is given is 7. The third group 1, that is a phosphotransferase with the hydroxyl group is the acceptor. So which group is accepting this phosphate? So this one, it indicates the group which accepts the group. So the hydroxyl group is given number 1. Hydroxyl group is assigned number 1. The fourth group, D-glucose, is the phosphoryl group acceptor. Here, it indicates the acceptor molecule. Okay, so like that, the 4-digit classification given to each enzyme, it gives a full description of that what type of reaction is occurring, then what type of, if it is a transfer reaction, what type of group is transferred, when it is transferred, which group is accepting it, then finally what is the molecule that accepts the group. So it gives the full detail of the type of the reaction. This one we'll do in the next class, the 6 classes of the enzymes. Just we'll stop here, and if you have any question, then we can discuss that. Yes, anyone, any question?